Hello everyone, welcome to our webinar. I am Florent, I'm the CEO of Neurolite. We'll be waiting for the last participants to join in. We have today participants from all over the world actually. We expected mostly participants from Switzerland, but we also have some friends from Greece, Saudi Arabia, from the US even, and Germany. So welcome to everybody. Since it's time, I will start introducing the presentation, and after we'll talk specifically about the topic of today, the Near Infrared Spectroscopy. So just a few minutes to introduce ourselves and the company. Neurolite is a company that is based in Switzerland, near Bern. We are a small distribution company of about 15 people, working in this field for about 10 years. Our passion is really to deliver products that we select from the best suppliers from all over the world and deliver them to our customers in Switzerland, providing also support and service and training. So the products encompass neurophysiology products like electromyography, evoked potential, EEG, sleep studies, as well as neuromodulation systems like magnetic stimulation, transcranial electrical stimulation, navigation, as well as neuroscience, which will be more the focus today with near infrared spectroscopy, but also EEG and eye tracking. And of course, all the accessories that everybody needs to perform the studies of all this. Our specialty is also, of course, to make the installations, the support and repairs in our office or directly at customers. So we have dedicated technicians trained for this, and also to perform the maintenance on the devices. What we already started for a few years ago is also to provide courses, like conferences on selected topics. But we also provide individual trainings on devices. So if you have a need on a specific device of ours or even a competitive product, but in the same field, then we are able to provide one-to-one education. And what we started now is webinars. We already have had some about evoked potentials in EEG. And today you will attend to the NIRS session. Finally, Neurolite is not only providing solutions here, we are also active outside of Switzerland with some humanitarian projects, like here illustrated, a project in Burma, where we provided also training and some device for intraoperative monitoring, and in Togo, where we provided an EEG device for a project there. So that's about it. To introduce shortly our company. Now I will leave the word to a speaker. Today we have Coralie de Brac, who is a postdoctoral researcher at the University of Geneva, and also an expert in NIRS with numerous publications in this topic. She joined Neurolite a few months ago, so very fresh, to support us in the research field specifically. So I leave the word to Coralie and wish you all a very enjoyable webinar.
So good morning, everyone. So as just said by Florent, I'm Coralie de Brac, and today I'm really pleased to present you the first NIRS Near Infrared Spectroscopy webinar of the Neurolite Academy. And just prior to start, I would like to say that if you have any question during the presentation, just writing in the chat, so the discussion or the Q&A, and I will answer all your questions during the Q&A session at the end of the webinar. So now you know more about us. We can go deeper in our today's topic, and starting with the NIRS basic. So NIRS is based on tissue transillumination. So in other words, NIRS is a huge torch light up the hemoglobin to see the oxygen that is contained inside. So why measure the level of oxygen? It's because muscle or brain, like us, when we do physical exercise, when they are more activated, solicited, they need more oxygen to work. So for instance, if I'm doing arithmetic tasks, or 3, 4, 10, for instance, my prefrontal cortex, the PFC, will work more, and for that, we'll need more oxygen. So for you in research, if you can guess the level of oxygen, the increase or decrease, you can link directly the brain activation to your specific experimental task. And the NIRS do not work with every light. It's a specific light, near infrared light, to really assess the oxyhemoglobin and the deoxyhemoglobin. And how the NIRS really work? For that, you need 2 optodes for optical fiber. One optode will emit the light, so the transmitter, and one optode will detect the remaining light. So that is a receiver. Why remaining? It's because during the optical path length, so the light go deeper in the scalp in form of banana shape, and in your measurement, the oxygen will be absorbed. The light, sorry, will be absorbed by the oxygen. And usually in research, the optode, so the transmitter and the receiver, are separated by 3 centimeters or 3 centimeters and a half in average, and in children is around 2 centimeters, enabling the light to go deeper in the brain around 1 centimeters, 1 centimeters and a half. Now that this technology, we can also move forward with the technique. So the NIRS has some inconvenience, if I can say. It's because with the optical path length that I just mentioned, you can just measure cortical activation. So you cannot target subcortical region, such as hippocampus or amygdala, for instance. So it's maybe an inconvenience, but there is a lot of other advantage that compensate this inconvenience. So for instance, it's a better temporal resolution than fMRI, so functional magnetic resonance imaging, that also measure the level of oxyhemoglobin and deoxyhemoglobin. It's poorly sensitive to motion artifact compared to EEG, encephalography, or other neuroimagery technique. And it can be also truly wearable and portative, and we will see that in the next slide later. So now you are all experts in NIRS. We can move to the different type of NIRS in the market. And prior to start, I would like to add that all the devices that I will present you can be combined with EEG, but also transcranial current stimulation, TES, and can be synchronized with this little remote right here. So pretty simply. So to go back to the different NIRS device, you have 2 types of device. The wearable one, the portative and wireless one, and the traditional fixed system. So we will start with the traditional fixed system, namely Oxymon. So Oxymon, with so as I just say, it's a traditional fixed system. With functioning with box staking, you can just target and assess one brain area, or you can assess whole brain, so all the brain simultaneously. And as the NIRS is poorly sensitive to motion artifact, you can measure the oxyhemoglobin of the brain, but you can also measure the oxygen level of muscle during physical exercise, so TSI, tissue saturation index. And an advantage of such device, it's fully compatible with MRI, with different kind of optode. Like that, you can combine the temporality of the NIRS, the temporal resolution, and also the spatial resolution in your final result and data. Let's moving now with a wearable device, starting with a Bright system. So the Bright system, it's truly portative and wearable. So you can do experiments in outdoor and ecological experimental condition with a free motion participant. You can use a Bright on adult, but also in infant. And an advantage of such device, it's you can save directly the data in the device, and you don't need the software, the NIRS software, namely Oxysoft. And so to stay close, at least at around 150 meters to your participant with the dedicated computer. So your participant can run away, can bicycle, can go home with the NIRS, and you can get back the data further time at the lab with a key, USB key, for instance, or hard drive, external hard drive. Now we have the Oxymon system. So it's the same principles than the Bright, so outdoor condition, adult and infant, but here you have 8 channels, and you cannot save directly the data on the device. You need the computer. And finally, the Portalite and Portamon system. So it's 2 types of mini NIRS devices that can be really valuable also in research. So you can do also outdoor and ecological condition in adult and infant, even in young baby. But with this one, you can measure the oxygenation of the brain, but also of the muscle in physical exercise, for instance. So now to illustrate what I just say for the different type of devices, I will show you a different video that illustrate my word. I think you cannot hear the noise, but anyway, I will talk during the video to comment, and it was just chill music, so you don't miss really huge stuff. So let's go to an example of how to set up the Bright system, so the wearable one. So here the participant can simply adjust on his own the NIRS cap on NIRS N-band. So it's really a huge advantage comparing to EEG and many time to set up the cap. And second time, you just have to push a button to turn on the Bright. And if the Bluetooth connection was not already on when you push the button, you can already push the Bluetooth connection to connect with Oxysoft, so the computer software of the NIRS. And here, Oxysoft recognizing the Bright system, enabling to connect. But you can also turn on the Bluetooth to do the offline measurement. Here you see the optod template of the optod with the transmitter and receiver position and the online measurement. If the signal is not good enough, you can gently remove the air from the hole of the cap, the NIRS cap, to obtain a better signal, less artifact. And you can also adjust the pressure of the optod on the scalp to the participant also to gain in a better signal. The participant perform a cognitive task, and you see on the right, just here the data in real time on the 3D template. And an example of how the Bright can be used in physical exercise here in running. So that's it for the price. Let's do it exactly the same, but for the Oxymon, sorry, the fixed system. Like that, you can compare. So here on the left, you see all the boxes that are possible to do the maximum of channels, maximum of measurement. You can choose your NIRS cap. And again, the participant can adjust the NIRS cap on himself or herself. And you as experimenter, you just have to click the optod on the NIRS cap. And also, if needed, you can remove the air from the hole of the NIRS cap. This is a transmitter and receiver. Here, all the different channels during an online measurement. The participant now do a cognitive task. The online measurement. Now the participant has another N-band for another cognitive task, a custom one. You see better now the N-band. And now the experimenter is adding a trigger event directly on the Oxysoft software. But of course, you can do that also on MATLAB or ePrime, for instance. That is the Polymus. Polymus is a 3D digitizer to digitize your optod on a 3D brain template and to have the MNI coordinate of your transmitter and receiver. So here you see the position of your optod in a 3D template. And now it's not Oxymon anymore, but Octamon, one of the wearable device, the 8-channel one. Another video now to show you how to do or how you can combine NIRS. Here it will be the Portamon devices on muscle with other physiology or neuroimagery devices. So with this video, the NIRS is combined with ECG, electrocardiogram, and EMG, electromyogram during physical exercise, running. You see the experimenter and also preparation with the Portamon devices and the Oxysoft software. So they fix the Portamon device on the leg of the participant with only a little strap. So quite easy also. And they connect the EMG and ECG on the participant as well. So you see here the Oxysoft software with all the measurement of the device in the same time with the participant freely running. So the computer and the software. And you see better now that on the top you have the NIRS measurement and above the ECG and EMG. Still in Oxysoft. And finally, the icing on the cake, if I can say, is to use Portamon, so on muscle also that you just said, but underwater. So I left you with these beautiful images. Few seconds. So now they adjust the Portamon under the swimming cap of the swimmer before to go underwater again. Here an illustration of the hemoglobin transportation. And now it's a tube Portamon, but in waterproof plastic bag that they fix on the leg of the swimmer now. with again a simple strap. So here's the score of the team. Unfortunately, no Switzerland flag here. And in real time now, you have the TCI, tissue saturation index, during the swimming competition. With also the hemoglobin illustration. So that's it for the product and the demo. And now to show you how really to use the NIRS in research, to give you some example, I selected a few papers recently published in peer-reviewed journal to give you really an overview, a quick overview of how the NIRS can do in research. But obviously there is a lot of other application that's still possible. First, functional NIRS, functional studies. So with this paper that I published 1 year ago now on the human recognition of emotion and pseudo word. For that, I use the Oxymon devices, a fixed system with 8 channels on the inferior frontal gyrus, IFG. So close to the frontal cortex, in the frontal cortex actually. And what I found, so quickly, I will not enter all the time into detail in the different results because it's not the topic today, but that you can understand clearly what I mean. It's here. Sorry, you have the mean value of the oxyhemoglobin concentration. Here the different emotions, different vocal emotion, anger, fear, and neutral. You have also the left and right hemisphere, so left and right IFG. And the dark color represents the emotional condition. So the participant had to recognize emotional vocalization. And in lighter color, the contrast with the word, they had to recognize the word and not the emotion anymore. And you have also the task, the recognition task, or you have to recognize, categorize the emotional word, or you have to discriminate the emotional word. And overall, what you see that the left and right IFG is modulated by the interaction of all these factors. And overall, for the NIRS, the meaning is that the NIRS can assess really thin processing, such as emotion, in a peculiar brain area, such as IFG, and in a pretty complex paradigm. The second publication is about functional connectivity. As fMRI, you can do also connectivity with NIRS. And for this purpose, Vergot and collaborator published this paper in 2017 and developed a method for the time frequency effective connectivity. For that, they use both simultaneously Oxymon and Octamon in prefrontal cortex, but also motor region. And what they found is that there is a connectivity between the frontal region. There is also a connection between the motor region, and interestingly, there is a connectivity between the frontal region and the motor region in the left hemisphere. It will be also interesting for you to combine the NIRS with other neuromodulation devices, such as the transcranial direct current stimulation, TDCS, as with this paper of Mutalib and collaborator, also published in 2017. For that purpose, they use Oxymon on motor and frontoparietal cortices, on the left one, and they use also TDCS, so the StarTIP system, with 4 electrodes also on the left motor cortex. And what they found. So here you have the level of oxyhemoglobin concentration. In dark and gray, you have the TDCS session, so the first one and second one. And in white, it's a sham group, so the participant out of fake session of TDCS that was turned off, so no session of TDCS at the end. And what the author found is that for the 4 regions that surrounding the 4 electrodes of the TDCS, there is an increase of oxyhemoglobin during the TDCS session comparing to the sham group, or there is no TDCS session. But of course, the participant didn't know that. In combining devices, you can also combine neuroimagery devices such as EEG and MRI, concurrent FMRI. It's the topic of this paper of Envar and collaborator published in 2016. In the blue dot or purple one, depending what you see, it's represent the electrode, the EEG electrode, and in green and red, the receptor and transmitter of the NIRS. And what they found, so the results are a little bit complicated, but I will explain you. So the Granger causality, it's a statistic that assesses the causation. So here it's if, for instance, the motor cortex drive the activity of the frontal region during the specific task of this paper. And here the task was a finger tapping task, a simple finger sequences and a complex finger sequences. And that they did so. The graph of Granger causality for all the tasks, for all the devices, so here the fMRI, the FNIRS, and the EEG, sorry. And the colorful dots represent the different pathway between the brain region. And overall, the author demonstrates that there is a bidirectional connectivity between the somatosensory network during the finger movement task. And finally, the last paper, but not at least, is the NIRS can be used on other species than human, than us. The answer is obviously yes. So for this purpose, I published a proof of concept 1 month ago to use the NIRS on baboons, non-human primates. For that, I use Portalite, 2 devices of Portalite, one on the right hemisphere, one of the left hemisphere simultaneously on the motor cortex of the baboon or the temporal cortex of the baboon. And what I found, so here you have the AQ. AQ, it's asymmetry index. So we start with this kind of statistic. You can know if your brain activation are more lateralized in the right hemisphere, so here with a positive value, or more lateralized in the left hemisphere, so with the negative value. And that for all different channels, 1, 2, and 3, my measurement with a Portalite, and all my subjects, my baboon, Talma, Ruby, and Chet. And what I demonstrate in that for right and left arm movement or right and left sound that they were displayed in an earphone in the right or left ear of the baboon, there is contralateral activation. So if I move the right arm of the baboon, the left hemisphere was activated and vice versa, and exactly the same for the sound. If I display a sound in the right ear of the baboon, the contralateral hemisphere will be activated, so the left one. That is really well known in the human literature, but it was the first time that it was demonstrated in non-human primates with NIRS. So it will be good for the future that open a new perspective for the NIRS also in non-human primate research that will hopefully drive more non-invasive neuroscience in primate research. And now to wrap up everything, to wrap up this webinar. You should remember that NIRS is a non-invasive technique. It's truly wearable and wireless, if you wish, obviously. It's fully sensitive to motion artifact, enabling outdoor ecological condition. That is a huge advantage, I think, in research. You can combine the NIRS with other neuroimagery or physiology device, e.g. TDCS, TMS, MRI, EMG, etc., etc. You can use the NIRS also to assess the level of oxygen in the brain as well as in the muscle. And you can use the NIRS in a lot of different type of experimental research or fundamental research, such as EPR scanning, functional, infant, diagnostic, virtual reality, BCI, non-human animal, etc. Sky is the limit. So all these advantages with the NIRS can explain why the number of publications, so published in peer-reviewed journal, are exponentially increasing since 20 years now. On that, I leave you with a slide before our Q&A session. You can, of course, after the Q&A session, reach me if you have any question by email or by phone about NIRS, but also other neuroscience device that are represented here in our really cool brochure that I can also send you by email. And save the date for your next webinar on lung, also ultrasound next week. And also save the date the 6th of May for the next webinar on NIRS. And you will choose, the topic of this webinar. I will share with you now a little survey, and you will tell me what type of topic you want to discuss next time. I leave you a few minutes to fill out the survey, and I will share with you the result. I leave you 30 seconds more. Okay, guys, so I will show you the result, and that is a little bit tricky. We have exactly the same result for NIRS and infant, concurrent NIRS with other technique, and NIRS in outdoor. So I suppose for the next webinar, I will try to do everything. I will do my best, I promised. Here you can see the results, so 33, 33, 33. So thank you for your vote. And also you will receive a little survey after the webinar by email to have your feedback on the today presentation. So feel free to say what truly you thought. It was my first webinar, so be gentle, please. And on that, let's do it the Q&A session. And thank you very much for your attention. And I'm really happy to answer all you questions that you have. Just write in the chat and I will answer. Thank you. So, okay, so could you please explain the possibilities of NIRS in hyper scanning? So hyper scanning, it's to assess brain activation in 2 or more participants in the same time. So the good thing with NIRS is because it's poly sensitive, it can be wireless, so poly sensitive to motion artifacts, sorry. You can easily use, for instance, it's create more and more use in research on interaction, vocal interaction in human, for instance. So one of the mom head and one on the children, and to see the connectivities or interaction between both, for instance. And I'm actually trying to do that as well. Do you have any other question? Apparently no. Or I can see it at least. Okay, so if you don't have any other question for now, at least, I will end this webinar. And again, don't hesitate to contact me by email or by phone if you have further question later. I will be really happy to answer. So thank you very much, everyone, and hope see you the 6th of May. Bye-bye.